everyone, and welcome to AP Help 101. So, today I'm going to be talking about cellular respiration, and this comes a lot on the AP test, so let's get started. Let's just do a brief introduction about cellular respiration. We'll go more in depth in these topics. So basically, cellular respiration is when cells get energy that is stored in food and they move that energy into ATP. And there are two types of respiration. We have aerobic and anaerobic. Aerobic respiration is when oxygen is present, and anaerobic respiration is when oxygen is not present. And the presence of oxygen really makes a huge difference in the track that the cells take to create energy. There are two stages in anaerobic respiration: glycolysis and alcohol fermentation or lactic acid fermentation. And there are three stages in aerobic respiration: glycolysis, Krebs cycle, and the electron transport chain plus chemiosmosis. So let's talk about ATP a little bit. Adenosine triphosphate is what it is. It has a ribose sugar, 3 phosphate groups, and the adenine nucleotide. When one phosphate group is reduced, the molecule becomes more stable and it releases energy. And that's really important to understand, is that's how we get our energy from ATP. It turns from ATP to ADP, adenosine diphosphate, and that's when the energy inside it is released. ATP provides the energy for all cellular activity, and that's what makes it so important. So this is just a diagram of ATP. You can see the 3 phosphate groups on the left. On the right, you have the adenine nucleotide, and right in the middle, you have your ribose sugar. The structure of the mitochondria. You may think it doesn't play a huge role, but it's important to know at least. It has a double membrane. The outer membrane is smooth, and the inner membrane contains folds called cristae. The Krebs cycle takes place in the matrix, which is in the middle of the mitochondrion. And the electron transport chain takes place in the cristae. So here's just a picture of the mitochondrion. You can see the matrix is the stuff right dead center of the mitochondrion, and that is where the Krebs cycle takes place. And you can see on the inner membrane the folds, and that is where the electron transport chain takes place. So let's talk about glycolysis, as it is the first step in anaerobic and aerobic respiration. So in glycolysis, we break down 1 molecule of glucose into 2 molecules of pyruvate and 4 ATP. Now, since glycolysis requires an energy investment of 2 ATP, we only gain 2 ATP, so what we count as a gain is only of 2 ATP. It takes place in the cytoplasm, and it does not require oxygen. That's why we can use it in anaerobic respiration as well. Glycolysis provides the raw material for the Krebs cycle in aerobic respiration and fermentation for anaerobic respiration. So right now we have a total of 2 ATP being created in glycolysis. It's actually 4 created, but remember, we still use 2, so we only gain 2 out of glycolysis. The Krebs cycle is also known as the citric acid cycle, so you may see it written both ways. The Krebs cycle continues the oxidation of glucose. Now, there are 2 cycles in the Krebs cycle, so it goes once and twice. And each cycle generates 1 ATP, therefore the 2 cycles generate 2 ATP. And the Krebs cycle is an example of substrate-level phosphorylation, as well as glycolysis. Glycolysis, too, is an example of substrate-level phosphorylation. Now, the pyruvate bonds with coenzyme A to create acetyl-CoA, which is necessary to start the cycle. So before we can even get to the cycle, we need to turn that pyruvate into acetyl-CoA, or else we have no ticket into the cycle. And without the Krebs cycle, cellular respiration cannot continue. The Krebs cycle releases 2 ATP, 6 NADH, and 2 FADH2. And NADH and FADH2 are electron carriers, and we will discuss those further. So right now we have a total of 4 ATP, 6 NADH, and 2 FADH2. NAD plus and FAD. They are coenzymes that carry protons or electrons, and they are the oxidized form of NADH and FADH2 respectively. So basically, these guys are electron or proton carriers, and they play a huge role, especially in the last stage of aerobic respiration. Actually, in both types of respiration, they play a huge role. The electrons that are used that are carried by NAD plus and FAD. They are used in the last stage of aerobic respiration and also in anaerobic respiration, and they are used to create the most amount of ATP possible. The electron transport chain, also known as oxidative phosphorylation. So there's a proton pump in the mitochondria in the inner membrane, and the flow of electrons from NADH and FADH2 down the chain releases protons into the intermembrane space, which is in between the inner membrane and the outer membrane, hence the name intermembrane space, from the matrix, which is where the Krebs cycle takes place, making a proton gradient. The electrons come from NADH and FADH2 from glycolysis and the Krebs cycle. So basically what's happening here is that there's a long chain, and these electrons are going down the chain, losing energy each time it goes down the chain. It's like going down steps, but they lose energy each time they go down the step. So, when they lose that energy, a hydrogen ion is pumped across. So, from the matrix to the intermembrane space, a hydrogen ion is pumped each time the electron goes down the chain. So, we still have our 4 ATP, but by this time, the NADH and the FADH2 probably would have been used. Chemiosmosis. This is the second part of the last stage of aerobic respiration. This is where most of the energy is released. The hydrogen ions in the intermembrane space go through ATP synthase channels, which generate energy to turn ADP into ATP by adding a phosphate group. So these ATP synthase channels, they work like turbines. They are like turbines. These hydrogen ions in the intermembrane space, they go in through these ATP synthase turbines, and they start spinning, and then they put the ADP and a phosphate group together, and bam, they form ATP, which is what we wanted to create. And oxygen is the final hydrogen acceptor, and it creates the waste product of water. So we create 32 through 34 ATP in chemiosmosis, and you can see how this releases the most amount of energy and how important this is. And this is also an example of oxidative phosphorylation. So now we have a total of 36 to 38 ATP, and we have no electron carriers left, and the cycle continues. So now we're going to talk about anaerobic respiration, also known as fermentation. People sometimes just call it fermentation, even though fermentation has its own part, really. There are 2 steps. We have glycolysis and either lactic acid fermentation or alcohol fermentation. And remember, this does not require oxygen to create ATP. But there must be an adequate supply of NAD+. And that's important because since we don't have oxygen as an electron carrier, there definitely needs to be a good amount of NAD+ to carry electrons to create the ATP. So there are 2 types of fermentation, I said. There's alcohol fermentation, which turns pyruvate, which is created in glycolysis, into ethyl alcohol and carbon dioxide. And humans do use fermentation, but they certainly do not use this type of fermentation. This type of fermentation may be used by bacteria, maybe by some plants. Humans, however, do use lactic acid fermentation, where we turn the pyruvate into lactic acid, also known as lactate. And human skeletal muscles carry this out whenever there is not enough oxygen going to the muscles. So that's usually when we're working out really hard, and there's not enough oxygen going in to supply our muscles, so they turn to anaerobic respiration, and they start doing lactic acid fermentation. And actually, the pain that you feel in your muscles after you work, when you're working out, and that pain that you feel, that's actually lactate building up in your muscles. So that's it that you need to know about cellular respiration. If you have any questions or comments or suggestions, leave them down in the comments below, and I'll see you guys next week.